so it's great. There's a lot of really colorful banner art around, and you may wonder why there is nobody here. Today is Monday, and the diehards are the ones who are coming to this festival at this point. The vendors, the people who are coming for classes, teachers, professors, I'm sure. Sure, I'll take a bus. Thank you. Thanks very much. So I'm here at the Ion Torrent bus. This is by Life Technologies. I've actually just gone through the bus itself, but I just wanted to give you a little bit of a look around on the outside of the bus. It's kind of neat. It's got canopies and it's like a bus tour. Hello.
Hi.
Who are you?
So this is the Ion Torrent bus. I'm Mark Mooney. I'm the product manager for the Torrent browser software. And so what we have here is a small mobile sequencing laboratory. This is a small mobile sequencing laboratory. What we're doing here is demonstrating Ion Torrent technology and instrumentation. And we come on here. We can actually do sequencing on the bus from start to finish.
What kind of sequencing?
DNA sequencing.
Really?
Yes.
Oh. And how does that apply to?
Surprised.
How does it apply to Convergence?
Convergence of.
Oh. Are you here for the Convergence Festival?
No, I'm here for the International Society for Molecular Biology.
Oh my gosh, that's awesome! I like it! Totally cool.
But I'm happy to talk about Convergence, although I know nothing about it.
Oh, Convergence is a fiber festival. So I was like, a sequencing? Well, is it sequencing of stitches?
No.
This is so awesome! I love it.
So this is
May I look anyway?
Oh yeah, of course.
Okay.
Let me explain it. So there are two instruments that do sequencing. And so basically in the year 2000, the Human Genome Project was finished, and it took about 10 to 20 years depending on how you mark it, and it took hundreds of millions of dollars to complete one human genome, or two human genomes. And so what we're doing today is we can sequence a human genome in a day and produce all of the information. You look skeptical.
No, well, I'm just impressed.
So there's been a massive change in the speed with which you can produce sequence information and the speed with which you can analyze it as well.
Wow. And is it usually distributed in this form via a mobile lab, which is really what you have here, right?
This is an idea of like creating a mobile booth. Okay, so if you want to demonstrate your products and you drive to shows, and you have like something like the Partridge Family where you look out the window and wave.
And be in an air-conditioned space at the same time.
Yeah. So it's a way of looking at how you can just do sequencing very easily. If I can do it on a bus, then I can demonstrate this fairly easily.
Sure. So what are these pieces of equipment we have here?
Okay, so this is the instrument that does the sequencing.
Okay.
The underlying technology is here. So these are microchips, the same microchip that you have in your camera.
Oh dear.
Don't worry, I'll be reverse. The microchip that you have in your camera that you're taking this video with is something similar on this chip here, where it's a silicon wafer with underlying transistors, and the silicon wafer has tiny wells drilled into it, and those wells each take in one bead, and that bead is then sequenced in a little tiny well in that reaction vessel. So we have a strand of DNA. It's like a chain of bases, and those bases are GATC.
Okay.
So you add a base each time to your template strand, and then you register the pH change that happens when you add that base. You eventually create a trace of multiple wells producing a sequence.
And how long does the process take?
So running the chip takes about 2 to 4 hours, and analyzing the data 20 minutes to 4 hours.
So the process is this: how you want to sequence my DNA? What would you do?
So we take a swab from the inside of your cheek.
Okay.
We go away and we make a library, which would take about 4 hours. Just taking your DNA and cutting it into small pieces, and then putting it into some flanking sequence that we can recognize the sequence from.
Okay.
And so then I put it into the library. I bind the library to beads, to tiny microbeads.
Okay.
And I'd mix them up in a tube and amplify them with VCR, and then I'd take pads to show you.
So yeah, that's
Excuse me.
So pipette.
Using a polymerase.
you have to sequence the other strand.
So you synthesize the strand and then basically from the DNA synthesize the strand.
Right, you synthesize this
Take
one
the pipette,
once.
I take a tube and I pipette your beads up into this, and then I would apply it to this side of the chip like that. and the solution would run over the surface, and the beads would fall into the wells. Each bead is carrying a DNA sequence attached to it. Okay. So then you have this whole surface, and it's like like the chinko. It's like rolled a ball fallen into a well, and then there's a sequence attached to the bead. So let's take this.
Okay.
I mean, it's still 99% accuracy, but it's not 99.9. So
put it.
By the time you're back there, the accuracy drops a bit, and most people are comfortable.
That's a bit right.
So what you do is you can sequence the other way that way, such that
it fits only one way into the reaction.
Okay.
So now you have a high accuracy.
And then you close it down like that. And now you can drive liquid over the surface of the chip. So GATC.
GATC, okay.
Your bases. So each time you drive one base over the chip, certain wells will incorporate that base, and they'll have a pH change, and that will light up, and you'll see it as a set of signals that look like this.
Same sense going that way.
So you'll see this kind of tracing happening from each well.
Okay.
And then you eventually build up reeds. In this case, the reeds are 100 base pairs long, on average.
100 what?
100 base pairs. So
Base pairs, okay.
100 letters attached
Okay.
to the line.
Okay.
So then that's produced a sequence, and so one well produces one sequence.
I see.
And then I take all of the sequences. So this experiment, so what I'm looking at here is a report from an experiment that we did on the instrument earlier.
Okay.
So it produced 40 megabases, which is 40,000 base pairs, and it produced 399,000 reads. Okay? Our mean read length was 100 base pairs, the longest one was 203 base pairs. So then I can take software and I can map this to the genome that I sequenced it from.
I see.
And the reference genome here is human,
I see.
version 19.
I see.
And I map it to that, and then I get reads the map. You can ignore the barcoding bit. So this is the chip here. It's a picture of the chip. So this chip looks like that when you look at the beads that are on that surface. So the sequencing surface is here. And all of the yellow and red places are where you actually loaded beads effectively onto the chip. And this one didn't load so well, so it's around 40% the majority of bead loading.
I see. And that's just where the liquid landed.
Where the bead
landed. Okay. So they didn't fall into wells here.
Yeah.
This was
so
empty.
that's particularly a poor area in something like this as well.
Okay. Oh. Very cool. Total addressable wells 1,262,523, and each well you said
It's one bead, which makes one reed.
One strand, which is 100 to you have to 203.
Yeah.
Yeah. Oh, okay.
So it's basically, if I took your genome, I would have cut it up into very, very small pieces, and then read the very small pieces, and then quilted them back together.
Quilting. I like quilting.
So,
I gotta have a convergence reference. Well, okay, this is awesome. And then, so the human genome. So I'm looking at this map, right, of what would be the Homo sapiens genome sequence. And is that always the same for everyone? Like it's either this one in this position on or off, this one in this position on or off.
Everybody's genome is unique and individual, and so you take a reference, because we're all quite similar to one another, but we have variations. So my hair color is blonde, yours is black, and then the genes that control that are different. So we have the same genes, but they have different expression patterns or different ways of very subtle changes between them that produce one kind of pigment or another.
Right.
And so you can look at the changes and see them through the sequencing. So what you do is you take the reference, and then you align all of your reads to it, and then you say, how do I differ to the reference? What do those differences mean?
Okay. It's very helpful. I'm obviously still confused because it's a lot of information all at once, but it's awesome. I'm so thrilled I found you.
You're welcome.
Thank you. And I'm Trish.
Hi, I'm Mark.